Hi there, welcome to part 5. In this video, we're going to learn all about nervous tissue and really what makes up the spinal cord. So previously in this class, we learned all about neurons. We learned about how they are stimulated, they depolarize, they generate action potentials, they release neurotransmitters. So that action, those functions, make them the functional unit of the nervous system. Okay, so the nervous system, an organ system, lots of different organs. We have nerves, we have the spinal cord, we have the brain. Um, but we also have nervous tissue, right? One of four types of tissue: epithelial, connective, muscle, and nervous. And so neurons are by far not the only type of cell that is within nervous tissue, right? Making up these organs, the spinal cord, the brain, and the nerves. So neurons are the functional unit, and they need really specific requirements, right? They need to have just the right amount of sodium and potassium in the extracellular space. As soon as you release neurotransmitters, we need to start kind of like rooming them up. We need some protection from invading microbes. Right? So there are a lot of other cells within the nervous system that kind of support the neurons. Collectively, these supporting cells that provide physical and chemical support for the maximum functionality of the neurons, collectively they're called the glial cells or the neuroglia. Okay, so we are going to walk through this image down here in the corner. What we are looking at is just a little piece of the spinal cord. Note that in this section, we have part of the central canal, which is right here. We have some gray matter, which is here, it's a little bit darker, and we have some white matter. Okay. So let's walk through each of these different cells. You should be prepared to label these cells and to identify their functions as well. Okay, so the first type of glial cell that we're going to talk about are the ependymal cells. Okay, so these cells are forming the epithelia, the lining of the central canal. Okay, note that these cells have little cilia on them. Okay, so what they do is they actually circulate the cerebrospinal fluid. So just like we talked about earlier this semester, cilia can kind of create a current. They can push fluid along. We talked about that as our mucus escalator, so taking mucus from within our lungs and pushing it up and up and up so that we can swallow it and get it out of our lungs. So same kind of thing is happening here, just not with mucus. This is with cerebrospinal fluid. And so the little cilia are going to create a current circulating that CSF through the central canal, as well as kind of pushing it out under the arachnoid mater. Okay? So the lining of the central canal is made of the ependymal cells. And they also monitor CSF, for different signals, for different waste products, etc. Microglia, I like to think of these as the central nervous system's Roombas. So here's a microglia, lots of little extensions. And so essentially what they do is they act like the nervous system of the nervous system's presence within our central nervous system. So they're actually relatives of other white blood cells. So they're related to what are called monocytes and macrophages, or big eaters that just go around and like munch up anything that doesn't look like it belongs. And so they are very close relatives, and they have fairly the same function within the central nervous system. So early on in our development, they migrate into the central nervous system, and they remain as phagocytic cells. Remember, phagocytosis is engulfing or eating stuff, right? So if a bacteria makes its way across the blood-brain barrier, if some proteins have broken down, if you got a concussion, and so you have a lot of severed neurons within your brain, these microglia kind of come along and they clean up the mess. Okay. Astrocytes, right? Astro meaning star, cyte meaning cells. So these are star cells. These, again, lots of extensions, are the astrocytes, and these guys have so many different roles. First of all, they maintain the blood-brain barrier. So here we can see a capillary, so bringing oxygenated blood into the brain. Of course, we don't want just like anything that is in the blood to cross over that capillary membrane and get into our brain. So we have these astrocytes that kind of link together and make a relatively impermeable barrier between the blood vessel and the rest of the central nervous system. So they let things like oxygen, they let things like sugar come into the brain, but not chemicals, not poisons. Unfortunately, they make it difficult for some medication or most medications to get across the blood-brain barrier as well. And so treating neurological conditions can be pretty challenging because of this extra protective measure um, of the astrocytes. Um, Astrocytes also provide structural support, right? So here we can see an axon and some of their little feet kind of wrap around it and stabilize the neuron. Okay. Astrocytes regulate what is in the extracellular fluid, again, making sure that there's just the right amount of potassium and sodium and calcium. There's just the right amount of oxygen, etc., within this extracellular space. Okay. They also absorb and recycle neurotransmitters. So not only is there, for example, acetylcholinesterase that is kind of sweeping up the synapse um, after exocytosis, but astrocytes are also going to regulate that as well. Okay. Also, any kind of damage to the central nervous system is largely, like the healing process is largely regulated by astrocytes. Oligodendrocytes. These are the central nervous system's version of a myelinating cell. Okay, so we'll talk about what exactly that means here in a second, but oligodendrocytes, here is one of their cell bodies, and they have multiple extensions containing lots of fatty tissue, right? So lots of lipids. And these extensions wrap around the axons of multiple neurons. Right, so here is one part of the cell, here's another part of the cell, each one wrapping around an individual neuron, and essentially creating an insulation layer around multiple axons within the central nervous system. Okay. I've used this analogy before, I'll use it again. The axon in this case is kind of like the cardboard tube of a roll of paper towels, and this part of an oligodendrocyte cell wraps around the axon just like the paper towels would wrap around the cardboard tube. Again, creating a nice insulation layer. So what is the purpose of this? Myelination, right? So wrapping around this part of the cell with lots of lipids within it ensures that action potentials are really fast, and it makes sure they actually make it to the end of the cell. Because myelin, this fatty substance, is white, any place within the nervous system that has a lot of myelin is going to look white. Okay, so we can see here there's a lot of myelinated axons, and so this entire space over here is called the white matter. And over here, there's less myelin, and so it is gray matter. Okay, so I'll come back to that in just a moment, but before we get into that, I do want to elaborate just a little bit more on like what myelin is actually doing. Okay, so far in this class, we have talked about action potentials that are specifically designated as continuous action potentials or continuous conduction, right? So in an axon, voltage-gated sodium channel opens up, sodium floods in, depolarizes the next area of the cell, which then opens the next voltage-gated sodium channel, depolarization, opens the next one, and so on and so forth. So it's literally a chain reaction, one channel to the next channel to the next channel. Okay, there's no stopping, there's no jumping, it's just one, the next, the next. When we wrap our axons in a myelin sheath, right? Like with an oligodendrocyte, or in this case, this is a peripheral neuron. Each individual cell wraps around the axon like its own roll of paper towels. And so these little sections of the axon that are insulated with myelin, they don't have to go through this same process. An action potential in them looks a little bit different. Okay. And so what these little regions of insulation do, these myelin sheaths, is they increase the speed of the action potential, right? It's not this channel opens this channel, opens this channel, and every single channel has to be open. Instead, this insulated region allows the action potential to essentially leap across this insulated region and to the next part of the neuron. Okay, so the insulation increases the speed of the action potential. Okay, this is called saltatory conduction. Saltatory just means like leaping. So what we can see here is that when the voltage-gated sodium channels open and sodium enters, the sodium, the strength of that depolarization is maintained, right? So it's maintained through this entire section of the axon. Instead of dissipating, instead of sodium leaking out through sodium leak channels and the strength of that depolarization waning over time, instead, this really positive charge, positive 30, can essentially jump right across this myelin sheath, does not have to stop and wait for more sodium to flood in, right? Just shoots right across. And by the time it gets to the other end of the myelin sheath, there are more sodium channels to open up, re-strengthen that signal. Really strong signal leaps across this myelin sheath where it opens another channel and so on and so forth. And so the fact that you don't have to open every single channel essentially allows this action potential to go even faster than in continuous conduction. So let me try to show you a clip here. You have the link as well, and I highly recommend watching the whole thing. But what I want to show you is what happens with myelination, right? So in this demonstration, we can see that there are dominoes, right? When one domino falls over, it knocks over the next one, knocks over the next one. And so these are representing essentially depolarization of the cell and therefore opening of the voltage-gated sodium channels, right? In continuous conduction, every single domino has to knock over its neighboring domino. Every single one, it's relatively slow. But when you myelinate regions of the axon, it's a little bit different. Okay?
The dominoes can also be used to demonstrate.
Okay, so what we can see here is, hold on just a second, this is representing continuous action potential propagation. Okay, so one channel opens the next, opens the next. Here we have one sodium channel, a myelin sheath, one sodium channel, a myelin sheath, one sodium channel, a myelin sheath. Okay, so these are the little nodes, the little space in between the myelin sheaths. And so this teacher is going to send an action potential, right, down both of these axons, and we'll see which one reaches the end first. Okay, so yes, continuous action potential propagation is still lightning fast, but it can be even faster, and we can make it faster by insulating the axons. Okay. There are two different types of myelinating cells. One we already learned about in the lab. These are the Schwann cells. Each individual Schwann cell myelinates a single region of a single axon. In the central nervous system, we have many more neurons all packed together as tightly as we can. And in fact, we want to pack them together as tightly as we can so that we have space for more neurons. And so to save space, oligodendrocytes myelinate multiple axons at the same time so we can more densely pack them together. Okay, both are working in the same way. They're insulating the axons so the action potentials can jump from node of Ranvier to node of Ranvier. Okay. And so when we go back to our spinal cord image here, we can see that regions with a lot of myelinated axons, they look white. And in regions without a lot of myelinated axons, it looks a little bit more gray. And you'll see this in real life later this semester when we dissect the brains. Okay, so white matter regions of the spinal cord are where a lot of myelinated axons are grouped together. And so we can imagine that these are regions or these are tracts, they're bundles of fibers where action potentials are shooting up or shooting down to and from the brain. So white matter is a place where long distance messages are sent as quickly as they possibly can, whereas gray matter, without all these myelinated axons, instead it's more dendrites, it's more cell bodies. This is where the synapses occur. And we can even see that there are a lot of little synaptic bulbs all over this neuron. Okay, the gray matter is where synapses occur. So like short distance messaging, relaying from one neuron to another neuron. And we can see that there are a lot of different synapses showing us that this individual neuron, this multipolar neuron, is integrating the messages of many different neurons at the same time. Some might be stimulatory, some might be inhibitory, IPSPs and EPSPs. And so in the central nervous system, in the gray matter, reside the dendrites and neuron cell bodies of what are called multipolar neurons. And they're called multipolar neurons because there are multiple appendages, essentially, attached to or kind of branching off of the cell body. And so this model, right, this strategy is showing us that this cell is taking in a lot of different inputs, right? Many different cells are capable of communicating with these dendrites, and all of that information is integrated together, ultimately resulting in either, yes, there's an action potential, or no, there's not an action potential. And so these types of cells make sense in situations where we want a lot of information before we actually make a decision. And so multipolar neurons are more common in the central nervous system, where we have to integrate lots of different incoming stimuli. Interneurons, which are passing information from one cell to the next, right? And motor neurons, right? So we need to take in a lot of different information before we send a message out to the muscle cell to actually do its thing. Okay, so we don't want to jump the gun. We don't want to not have the full story before we say, yep, the muscle can contract. Okay. Also, in terms of motor neurons, there are many different reasons why we would want to contract a particular muscle. And so, all of those different reasons from all of the different parts of the brain and from, reflexes and everything, all of that information, or all of those different sources of information can stimulate this motor neuron to stimulate the muscle cell. Okay, so multipolar neurons are motor neurons, and they're just in the central nervous system. Okay, that is the end of this video. One more as part of this lesson. Thank you so much.